as you trickle back in from the break. My task today, this is a little bit of a transition talk, going from the overview presentations on the autism phenotype, the importance of heritability and genetics, to talking a little bit about epidemiology. So what I'm going to try to do is give a little bit of a framework of the way epidemiologists think about studying environmental risk factors. So, okay, here's my slide on complexity, and you know, this was we didn't pre-plan this, but you've heard every speaker so far has talked a little bit about the complexity of the autism spectrum disorders. I've got on my slide, I've got genetics, I've got environment, and I've got the phenotype over there to the right. I don't have neurobiology even on the slide, but we've heard a little bit about how complexity is the watchword when we think about neurobiology as well, and you know. When I talk about complexity to audiences that are a mixture of researchers and clinicians and other interested folks, you know, I start to get a little worried, you know, because is this the researchers doing excuse making here? Are we apologizing? We're saying, this thing is too tough, you know. That's I worry that it leaves people with sort of a negative feeling. You know, the notion is other researchers are saying this is complex. We're not going to get anywhere, and that's not really what's going on here. It's actually a positive thing that every presenter that you're going to hear today probably has a slide on complexity, because it shows that across the disciplines, from neuropathology to genetics to epidemiology, we're acknowledging and understanding that this is a very complex disorder. And you would rather have a bunch of scientists who understand the complexity and are trying to integrate it into their work than scientists who are thinking, oh, well, you know, this is easy. There's one gene, it's my thing, I'm a geneticist, and I'm going to find that gene. Or there's one lesion, and I'm going to find that lesion. Or me, an epidemiologist, there's one exposure, like cigarette smoking and lung cancer, I'm going to find that exposure. We all understand that that's not probably going to be the case, and that means that we're going to be getting ourselves oriented properly to do our work. So just a couple points on this. You've heard a lot about this. Over on the right, under ASDs, I have S in parentheses, which again shows that we're thinking about different types of autisms. DSM-4 represents the idea that we started with those clinical classification criteria that you heard about, which categorize kids as having autistic disorder or PDD, NOS, or Asperger's, and that's probably limiting. We can think about other clinical subtypes, so categories again, but maybe we're talking autistic disorder with cognitive impairment. Maybe we're talking PDD, NOS with associated gastrointestinal symptomology. So we have other clinical subtypes that we're going to start looking at. And then I also put social communication down as one potentially continuous phenotype, which is another way that we need to think about endpoints in this research. Not just autism yes, autism no, but maybe studying the distribution of these underlying phenotypes that are clearly important in autism. And then up at genetics and on the top, your right, I'm having the same problem orienting that Peter did, your top right, stage left. A couple of different genetic mechanisms that are probably involved. There probably are some putative risk genes that are passed on, but again, there are many of them, and maybe they're not so common, or maybe some are common with low penetrance. There's this issue of these new mutations, which are becoming more important. There's also the notion of teratogenic genes and epigenetic regulation, and I don't think anyone touched on yet. Teratogenic genes, and I'll talk about this a little bit more later on, are genes that maybe, they're not important because they're passed on to the child, but because they're genes that the mom carries, and they're involved in regulating developmental processes like the intrauterine environment during pregnancy. So those may be important genetic factors, but they're not important in that they get passed on to the kid. They're important in that the mom carries those genes herself. And then epigenetic regulation, this is sort of a whole nother world in understanding how the genome affects health. Not only the codes that are passed on or what's encoded in our genes, but there are also chemical modifications of those genes that can affect how genes are expressed. And these things can be modified by environmental factors. So thinking about complex genetics and how it interacts with the environment is going to be very important moving forward. And then lastly, under environment. So we haven't really started talking about environmental factors, and you're going to hear a lot from the speakers that follow me about specific environmental exposures. But studying the environment is also complicated. We can think about environment with a little e, and that's really everything that's not heritable. right? So that's everything from diet to lifestyle to sort of big E, classic environmental exposures that are xenobiotics, things like chemicals or infectious agents, things that we don't really need for life, but that we're exposed to anyhow. And also when we study the environment, we have to recognize that there are lots of factors that affect exposure. Social, physical, cultural determinants of who gets exposed at high levels and who does not. And also when we look at environmental exposures, we have to think about not only, again, simply, it parallels what I said about ASD, not only simply are you exposed or you're not exposed, but how much were you exposed to, for how long were you exposed, and were you exposed during the right etiologic period, the period where physiologically exposure is going to influence the outcome. So epidemiology is a little bit like pornography. I know it when I see it. I know what epidemiology is. But what is epidemiology really? So it comes from the Greek, and it's the idea. The translation is the study of what is upon the people. And when we think about epidemiology, so the people is important, and population approaches are critical to a definition of epidemiology. So we tend to lump epidemiology into descriptive epi and analytic epidemiology. Descriptive epidemiology is describing the frequency and distribution of the disease in the population. How common is it? Are there groups of individuals, old or younger, different racial ethnic backgrounds that have different prevalences or incidences of disease? And looking at those patterns starts to give us some early clues about what might be causing disease. And things like that Peter talked about before, looking in the CDC surveillance data and seeing whether or not frequency of disease patterns are different in different groups can give us clues about things that we want to study through more formal, more rigorously designed analytic epidemiology. When we talk about analytic epidemiology, we're talking about studies that use samples drawn from the population. And we estimate exposures between risk factors and outcomes by looking at whether the frequency of these exposures differ in different groups. So we can look at folks who are affected and folks who are not and see whether there are differences in the frequencies or levels of exposures in those two groups. Similarly, we can follow populations who are exposed and track outcome frequency in those groups and compare them to groups who are not exposed. But what's key here is studying the population, right? So what we try to do in epidemiology is get representative samples. There are lots of examples, and oftentimes science sort of starts with select clinical samples, right, and then moves forward from there. But those select samples can lead us astray sometimes. Again, if we think about smoking and lung cancer, which is a triumph for epidemiology, right? We've associated cigarette smoke exposure with lung cancer risk, obviously. Early studies compared cases of individuals with lung cancer to a convenient control group, which was hospitalized patients who were there for heart disease, because there was no suspicion that smoking was also linked to heart disease. So we didn't select a representative sample of all individuals in the population who could ultimately be at risk for lung cancer. We studied this convenient sample of folks with cardiovascular disease, and that made the smoking effects look smaller. So while we saw some interesting data from I'll just pick on the AGREE population on, say, prevalence of GI symptomology, we have to realize that AGREE families are very different from all families with autism. And it may be that, for some reason, prevalence of GI symptomology in that group, because they're multiplex, because they're an activist family group, they know what's going on, maybe that prevalence doesn't represent what it truly is in the population. So we need to get these representative samples, and epidemiologic studies strive to do that. All right, so I want to talk a little bit quasi-philosophically about the way epidemiologists approach analytic epidemiology. And this applies to other disciplines as well, I think. We have a set of studies that we think about as being hypothesis-driven analytic studies. All right, and this is where we have a specific idea about an exposure-disease association that we want to study. And ostensibly, these investigations are grounded in some a priori evidence. I mean, we had to have some reason to suspect that exposure A might be linked to autism, and some theory. And so we have a framework to set up the study. We've got some evidence, we have maybe some other studies that were done in epidemiology, maybe in other disciplines. So we have something to build on. So when we're building on something, we can think more critically about what other variables we need to measure to make sure that we're not seeing spurious associations because we're not controlling for other variables. We can think about what kinds of samples are going to be optimal for studying this potential association. So these hypothesis-driven studies tend to have a more rigorous conceptual framework around them because they're based in some prior theory and evidence. Now, the other kind of analytic epidemiology is what I will call more exploratory analytic epidemiology, and this is more of a discovery science approach. And these types of studies often get kind of a bad knock, and sometimes they're called fishing expeditions. They're not hypothesis-driven, they're fishing expeditions. But I want to point up that they can also be positively viewed. And this is the story about sort of looking under the lamppost. This idea there's an inebriated guy outside of a bar looking for his car keys. You've probably heard the story before. And he's looking under the lamppost, and somebody asks, or he's looking in this one area. So he says, "Why are you focused here?" And he says, "Well, this is because where there's light. His keys may not be under the lamppost, but he's going to look there." And so exploratory studies positively are viewed as something that may take us outside the box and may come up with some initial clues about mechanisms or exposures. Now, it tends to be more difficult to design rigorous exploratory studies because, remember, we're looking for novel connections. So we don't really know what the framework is. We don't know what other covariates to measure to adjust. And we also, when we're looking for these novel connections, we may have a pretty long list of things that we're interested in exploring. So when we look at potential associations between lots of exposures and an outcome like autism, we can do maybe a screen for 50, 60 exposures. The likelihood of having one association appear in our sample just by chance is higher. So you have to watch about false positive findings when you do exploratory epidemiology. But then what time am I done? I know you're going to give me the yellow slot. Okay. So there's going to be calls for more exploratory work related to the environment and autism. And this is a paper by Larry Needham from the CDC. He did a lot of seminal work in lead exposure and developmental neurotoxicity. And he's reviewing, looking at developmental toxicant exposures via biomonitoring. And in this paper, he talks, and I'll just summarize this text, he talks about taking an approach where we have affecteds and unaffecteds. We take samples, biologic samples. We use high-resolution chromatographic systems coupled to universal detectors such as time-flight mass spec, and we find a full scan of all eluting chemicals, basically everything that comes out of these biologic samples. And then we use statistical methods to sort of figure out what differs between cases and controls. This is clearly an exploratory approach. So the message I want to leave you all with when it comes to these types of exploratory studies, or really when it comes to any kind of epidemiologic studies, and actually really it generalizes to other types of scientific research too, is that the general way that this guy, that Reverend Thomas Bayes, if you've ever taken a statistics course, you've probably heard of Bayes' theorem. He came up with this idea back in the 18th century, and it's a mathematical formula that's used in different statistical methods, and it's very impactful right now in statistics. But there's a general way of sort of summarizing his thinking, and his thinking was when you develop some new evidence, and that can be from a study, that's part of what you need to make a conclusion from it. What you really are doing is linking it to a prior probability, and that's all the other evidence and hypotheses that went before it. So if you find some evidence suggesting an association between a chemical and an outcome like autism, there's really not much else suggesting that this is real or plausible. You're going to give that different play, different weight than you are if it comports nicely with theory and other types and evidence from other types of scientific disciplines. So try to keep this framework in mind as you're monitoring the epidemiologic research as it unfolds over the next 10 years. I think it's going to be very, very important. So what I want to do now is I want to segue from quasi-philosophic differences between exploratory and hypothesis-driven research and give you a little bit of an overview of some of the basic epidemiologic study designs. The first of these are ecologic studies. And these are studies that take group-level exposure data and group-level outcome data, frequencies of exposures in population groups and frequencies of outcomes, and correlate them. So we're not taking individual-level data on individual subjects. This is an exposed subject. This subject has the outcome. This is an unexposed subject. That subject does not have the outcome. We're just correlating rates in the community. And these studies tend to be exploratory, and you really have to use caution when you're interpreting these group level studies. When I teach my students in introductory epidemiology, I use this example, and it goes back to the 1930s, and it correlates stork sightings in the city of Oldenburg in Germany to the population. Right? Group level data on number of storks sighted in the city, and group level data on the population size. And it's a beautiful correlation. at the group level, right? But the evidence that this is providing on the causal association between storks and population is probably pretty weak. And we need to be cautious about studies like this. This is a study in the autism research domain that linked rates of autism in counties to precipitation rates in those same counties. So the dark, and this is in the state of Washington, the dark colored counties have higher autism rates on the bottom, and you can see there's nice matching to higher precipitation rates on the top. And the hypothesis that was built around this correlation was one of television watching being associated with autism risk. Kids in rainier areas might watch more TV. And so the theory went that might be correlated to autism risk. So, again, a lot of extrapolation from a group level correlation going on with that line of reasoning, so be cautious. And there have been other ecologic studies. This is one that's gotten a lot of attention, that's correlating autism prevalence rates measured through administrative data with rates of vaccine coverage, in this case for MMR coverage. And again, these kinds of ecologic studies need to be cautiously interpreted. We need to be certain that those trends in the rates are reflecting true risk, and those trends in the exposure are also reflecting true risk. So they're important studies, they're good studies, they can generate ideas, they can be good studies for generating ideas, but as far as causal evidence, we need to interpret them with caution. So next we have case control studies. So these are individual level studies where we have data on affecteds and unaffecteds, and we have data on exposures at that individual level. We know about the exposures to the affecteds and the exposures to the unaffecteds. There can be challenges in these because these studies are done retrospectively. We have kids who are diagnosed with autism, say at age 3 to 5. We have to go back and recreate what their exposures were, either by asking parents, looking in records. We're not measuring those exposures prospectively in real time. So this can introduce measurement problems which can affect our ability to draw good conclusions from these retrospective studies. Nonetheless, individual level data, which can be much stronger. We've been doing these kinds of studies for quite a while in autism. This dates back to 1979, a case control study on viral exposure in autism. This is a more recent study by Roberts et al. That looked at pesticide exposure and case control status of autism. Now the pesticide variable was interesting because they didn't measure, they didn't draw biologic samples from kids and measure for pesticide levels in the bloods. They linked it to a very nice database on agricultural pesticide use in California, and they linked it geographically to the address of the mother at the time the child was born. And they looked at a number of chemicals, used fairly rigorous methods for controlling for multiple comparisons, and were able to draw some conclusions about links between organochlorine pesticide exposure and autism risks. The graphs show, the bottom shows highest quartile exposure to organochlorine, and along the x-axis is time. So the 0, and I know this might be a little hard to see because the screens are a little bit small. The line is time of conception. The gray area is first trimester. So essentially those spikes show that there's a risk associated with organochlorine exposure in the first trimester window at high levels in the bottom graph. And then you can see as you move down, as you move up in the graph, third quartile exposure, second quartile exposure, first quartile exposure, there's still an association at third, and then it dwindles, so it shows dose response. Now there are other studies going on. This is the CDC Cadre SEED study. This is a very large case control study in 6 sites around the United States, 900 cases, 900 typically developing controls and controls with other neurodevelopmental impairments. There's going to be a lot of data collected on individual exposures, biomarkers, genetics, but again, retrospective, especially as far as looking at early life and in-pregnancy exposures. You're going to be hearing a little bit about another big case control study, the CHARGE study, which already has some data collected, and Dr. Hertz-Picciotto is going to be talking to you about that, and Cadre is a similar designed retrospective case control study. The other, now another design are cohort studies. These are studies where we monitor prospectively. Okay? So this maintains temporality relationship between exposure and disease, and it offers us an opportunity to measure those exposures in real time and do a better job measuring exposures. The other advantage of cohort studies, as opposed to retrospective case control studies is that those case control studies, you can imagine from the name, are very much tied to dichotomy, right, to cases versus non-cases. In a cohort study, you can certainly look at risk of discrete events in a cohort, but you can also look at those continuous phenotypes in your cohort and contrast whether or not exposures are linked to being further up or further back on a particular continuous phenotypic variable. You need large numbers for cohort studies, especially when the outcomes are rare, and they can take a lot of time and a lot of money. And sort of the poster child for a lot of time and a lot of money in a cohort study is the National Children's Study, some of you may have heard about, which is just starting now, where 100,000 pregnancies are going to be followed through age 21. These pregnancies are representative of the whole U.S., a lot of comprehensive exposure data collected in pregnancy and in early life. There are a lot of endpoints that are of interest here, both neurodevelopmental and also childhood asthma. And again, we're going to be following these kids through age 21. So a lot of diseases are going to be on the radar screen for the National Children's Study. So autism is on the radar screen, but the amount of effort that's being devoted specifically to following up for autism is a little less than is desired right now, and a number of groups are working to try to enhance that endpoint surveillance for autism spectrum in this cohort. There are some examples of, this is going to be a little hard for you to read, so I'll just tell you about it. There are some other cohort studies that are starting to take peeks at autism. This is a cohort, the Chamacos cohort in California, that follows about 500, 600 pregnancies in this area of California where there's high pesticide exposure. So they're following this cohort of pregnancies with high exposure, and they measured a lot of neurodevelopmental endpoints in these kids, including they used the CBCL, PDD subscale to get at autistic-like symptoms, and they did find an association between exposure to organophosphate pesticides in this particular study and autism phenotype as measured by the CBCL. Now, there were some inconsistencies with other neurodevelopmental endpoints, and this was a fairly small cohort, so there's some limitations here, but it was another cohort study, prospective monitoring of exposure during pregnancy and early childhood. So those are the classic studies, ecologic, case control, and cohort. But I think what we're going to be seeing in the next decade or so are studies that have more novel designs coming into play in epidemiologic research on environmental exposures and autism risk. We're going to see studies that are focusing on populations of special interest. And you've already heard about some of these populations already. The NICU group has been mentioned. And these kids who come from stressed pregnancies have an exposure profile on certain factors that's going to be very different from other kids. And I think it's going to be important to look at populations like this. The poster child for studies of special populations are the Dutch famine cohort. You may have heard of this if you've studied psychiatric epidemiology before. These are pregnancies that occurred during a time of extreme starvation post-World War II in the Netherlands. And there have been studies showing that linking a number of CNS events to being born during that time period. So I think we're going to be seeing more of this in autism research as well. We're also going to see studies that take advantage of extreme exposure distribution sampling. So if we have data on exposures for a large cohort, there are some clever things that we can do by just studying those at low doses and high doses to be able to help us with certain kinds of difficult to study problems like gene-environment interaction. And then there are also some special family designs that we can set up to study certain problems like gene-environment interaction. And this PENT study is one example where we look at trios plus also their grandparents' generation. And the last thing I'll mention are genetically enriched cohorts, where we study families that already have, we're going to be building a study, we're going to be studying pregnancies where there's already an affected child. And we're going to be monitoring those subsequent pregnancies very closely and following the kids prospectively for autism and other neurodevelopmental endpoints. So my time is up, so I'm going to skip through a few slides here and just end with these summary predictions. We're going to see more exploratory epidemiologic analyses of environmental exposures in the future. They're going to come from ecologic studies and from the large case control studies that are being fielded. We have to be careful consumers of this science. We're also going to see hypothesis-driven studies of environmental exposures following this. And hopefully these are going to be motivated by advances in other disciplines, neurotoxicology and neurobiology. And these novel study designs are going to be our best chance, I think, for embracing autism's complexity and finding ways of revealing connections between environmental risk factors and autism. Thank you very much.